Hello friends, welcome to Bhagati online classes. Today we are going to discuss the structure of DNA, that is deoxyribonucleic acid. Okay, the so-called DNA, that is bDNA, was proposed by F.S.C. Crick and J.D. Watson in the year 1953, for which they have been awarded Nobel Prize in 1962. Okay, so let's draw the structure of DNA, that is the double helical structure of DNA proposed by Watson and Crick. Okay, so here we can see the structure of DNA here, double helical structure of DNA. You can see the the two strands are in twisting condition, that is the helical condition, and here the two strands are running in opposite direction, that is they are anti-parallel in nature. Here you can see one strand is running one strand is running from 3' end to 5' end, whereas another strand is running from 5' end to 3' end, such that 3' face of one strand is facing to the 5' end of another strand. Okay, and here what happens, the core is consisting of nitrogenous bases. Okay, here you can see adenine bonded with thymine by 2 hydrogen bonds, whereas guanine bonded with cytosine by 3 hydrogen bonds. Thus, the nitrogenous bases are the core forming constituent particles. But whereas the sugar, backbone sugar, they are outer to the core, and here you can see that from the structure that here there is a depression and here another depression. The deep, this is the major depression, and this depression is called as major groove. Here it is major groove, whereas the small depression is called as the minor groove. What is that? Minor groove. Okay, and the length of minor groove is of 12 angstrom, whereas the length of major groove is 22 angstrom. That is why, and here on the other side also you can see this is the major groove, this is minor groove, and the total length of the both major group and minor group constitute 110 DNA. Okay, and the length of, because the minor group is of 12 angstrom and major group is of 22 angstrom, that is, and both these two groups constitute 110 DNA. That is why the length of total 110 DNA is 12 angstrom plus 22 angstrom, that is 34 angstrom. Okay, and here what happens, as adenine is always bonding, it is bonded with thymine by 2 hydrogen bonds, whereas guanine is bonding with cytosine by 3 hydrogen bonds, and this rule is fixed, and this is called as base pairing rule. Okay, and in case of DNA what happens, the amount of purines, amount of purines present in the DNA is equal to amount of pyrimidines. Okay, that is, here you can say purines, you know, adenine and guanine. Total amount of purines is equal to total amount of pyrimidine, that is cytosine plus thymine in case of DNA, and this is called as Chargaff rule. Clear? Okay. So next is here the distance between two nucleotide base pair or BP. Here you can see AT, the one pair of nucleotides, another pair is CG. These two are the successive nucleotide pairs, and the distance, as we know that in 1 turn DNA, from that is from minor group and major group, 10 base pairs present. How many base pairs? 10 base pairs present. That is why, and the total length of 1 turn DNA is 34 angstroms. So the distance between, or the length between any two consecutive or the successive nucleotide base pairs is 3.4 angstrom. Here you can see, okay, this is one base pair, another base pair, and the distance is being 3.4 angstrom. Clear? So this is the overall structure of the DNA. Okay, and here one thing that as these two strands are in helical mode, they are in twisting condition, and here the bDNA that has been proposed by Watson and Crick, okay, it is right-handed in nature, it is twisting to right hand side. Okay, and here the helical diameter is 20 angstrom. Okay, the helical diameter is 20 angstrom. Okay, so next we'll move on to the chemical structure of, or the molecular structure of DNA. Okay, and we know that the constituent particles of the DNA is nucleotides. Okay, the two strands are the chains, or the polynucleotide chains. Okay, each strand consisting of the many nucleotides. Okay, and you know each nucleotide is consisting of one nitrogenous base, a phosphate group, and a sugar molecule. Okay, and the sugar present, you know that in case of RNA it is, okay, what is that? In case of RNA, that is ribose sugar, but in case of DNA it is deoxyribose sugar. So that is why the name of the DNA is, that is deoxyribose nucleic acid. Okay, so now we'll go to through the structure. Here you can see this is a sugar molecule. Let us consider to the first prime carbon atom, what adenine group is added, and to the 5 prime carbon atom a phosphate group is added, and the next nucleotide, to the next nucleotide 3 prime atom of 3 prime carbon, or the member of this nucleotide, okay, another phosphate group is attached, which is again attached to the 5 prime carbon atom of next, okay, nucleotide. Here you can see, okay, this is 2, 3, 4. Okay, another you can see here, draw, it is adenine that is bonding with thymine, and it is, here you can say, phosphate group. Let us consider there is G here, C another. Okay, five phosphate group, here 5. Okay, so here this is 5 prime end, this is 3 prime end, here it is 3 prime end, and here it is 5 prime end. Okay, so the two strands are running anti-parallel to each other. Okay, and here you can see the core of the DNA is consisting of the what is that, nitrogenous bases. Okay, that is purines and pyrimidines, whereas the strands, the backbone, are sugar phosphate backbone. Here it is sugar, that is phosphate, and here the bond between two nucleotides, that is, this is called as phosphodiester bond. What is that? Phosphodiester bond. Whereas the bonds present between the bases are hydrogen bonds. What are those? Hydrogen bonds. And the bond present between here, okay, the bond present between sugar molecule and nitrogenous bases, one nitrogen base, that is called as glycosidic bond. What is that? Glycosidic bond. Clear? So this is the overall structure of DNA, that is molecular structure. Okay, so okay, clear? I hope you might have gained some matter thing from this videos. And to get new videos, to update with our new videos, you should subscribe our channel. Okay, thank you to all.